Video is about the basics of the gastrulations. So that just recall about what happened in the first 2 weeks in embryonic lives. So as we know, a bit recall when we have the ovulations, there will be the secondary oocytes will actually rest at the metaphase 2 in the mysis 2, and after the ovulations there will be 12 hours after that there will be a fertilization if there is a presence of any sperm, and the fertilizations will follow with the pronuclear state and become a zygote, and the first cleavage will occur 30 hours after the fertilizations, and this is 2-cell stage, and it's further differentiate into 4-cell stage and then 8-cell stage. So all of this happens in the ampular regions of the tubes, and after that it will further differentiate and become the morula, and morula 16 cells, and compaction happens and become 32-cell stage of also a morula. So after the compactions, so on day 5 it will become a blastocyst, and the embryo will get into the uterine cavity and start to hatch away from the zona pellucida on day 6. Hatch away and implant in the endometrium, and it's complete the implantations on day 13. So basically the video is actually about what happened in between this and also the 3rd weeks of embryonic lives. So what happened is that during the implantations, the cells will, for the blastocysts, will form a fluid filled cavity, and they will have an outer and inner cell. So this is an outer cells and this is an inner cells. Or you can see it's a become the cytotrophoblast, and this become the epiblast. The epiblast, I will color the epiblast in the blue, and the then the cytotrophoblast will be in the orange color. So further differentiations occur. Further differentiations, the epiblast will divide itself and become few layers here, and outlining the cytotrophoblast and forming a hypoblast. So this area surround we have the cytotrophoblast, which will invade further with syncytotrophoblast into the endometrium, forming the secondary villi, and then the epiblast here will differentiate and become more epiblast, and the lower part will become the hypoblast. So this will be the future primary yolk sac, and this will become the epiblast will form another cavity within itself, forming the amnion, and this is the hypoblast. So one of the things that I think always me myself missed out when I learned about embryology is that the formations of the extracoelomic mesoderm, the extraembryonic cavity. So what happened is that there will be a formations of extraembryonic coelom or cavity. So just catch here straight away. So what we have here is that the cytotrophoblast, and we're gonna have an epiblast with the amnion here, and we have the hypoblast, which will become the primary yolk sac. And at this point, there will be a mesoderm, extraembryonic mesoderm, which will outline the space in between the cytotrophoblast and also the endoderm and ectoderm. So this initially a solid plate, but later will become a cavity. So we have here the amnion, the epiblast, the hypoblast, and also the yolk sac. This structure is called extraembryonic mesoderm. This is the extraembryonic mesoderm, and this is the extraembryonic coelom or cavity. Right. So when we have this, what will happen later is that a neurulations and also a gastrulations. So neurulations basically is a process of forming a neural plate. A neural plate, a neural tube, and a neural crest. So what happened is that this is the ectoderm from the side. This is we cut the embryo, something like this. We cut it, and we'll see from here. We can see this cross sections, but here is actually a picture from lateral. So we have this view now. We want to see from the side. So neurulations, basically there will be a two membrane here. So here we have the bucopharyngeal mucosa or membrane, and here we have a cloacal plate. So neurulation starts when there is a formation of the folding of the ectoderm, So they're gonna always catch something like this. The ectoderm will fold and forming a primitive strix here, and it will start from the caudal to the cranial. It will move upwards to cranially. So there is a fold here, which we call a primitive streak, and there will be a primitive pit or a hole. Primitive. This is basically the result of a folding. This is a lateral fold, and this will be a folding of the notochord, the folding of the ectoderm. So this is the primitive streaks, and this will be the this will be the primitive streaks, and this will be the pit. So the notochord is basically the folding of this ectoderm, and it will move cranially. This is cranially, and also caudally, and this will form the notochord. Notochord basically will become the nucleolus of the intervertebral disc. So what happened in the neurulation is that there will be a folding of the ectoderm, and it will pinch off from the ectoderm, forming the neural tube here. So we have the neural tube here. We have the neural crest here. The neural tube will become the central nervous system, And the neural crest will become the peripheral nervous system. This will become the skin. Basically, this is actually an oversimplification of this picture. So what happened is that after the neurulations, there will be further influx of there will be folding and further influx of the ectoderm into the space in between the ectoderm and endoderm. So there will be more cells influx here. It will fold and influx and occupying this space. And later, it will form the intraembryonic mesoderm. So influx of the ectoderm and occupying the space in between this will form the intraembryonic mesoderm. So let's catch the whole thing so that it's become a complete cross sections. So now we have an ectoderm with a neural tube and also neural crest with the influx of the mesoderm here. So the mesoderms, as you can see here, there will be an influx of the mesoderm, and we have three parts of the mesoderm, which we have the first this catch this way because it's actually the connections between intra-embryonic mesoderm and also the extraembryonic mesoderm. So basically, the whole picture will be something like this. So this is the amnion up here. Amnion. And this mesoderm, it will become the somatopleuric mesoderm. And this will become the splagnomesoderm. So here, down here we have the ectoderm outlining. And we have a cavity here, extraembryonic cavity. So let's label the picture so that we can get a correct idea and also easy understanding for this topic. So right here we have the ectoderm. This will be a neural tube. And this is the crest. This is the amnion. This will be the extraembryonic cavity, oscillum. This will be the primary yolk sac. This will be the paraaxial mesoderm. This will be intermediate mesoderm. And we have two here. It's a somatopleuric mesoderm. And at the back there, down here, we have the splagnopleuric mesoderm. So the question that they always ask from here is that what is the origin of every single part of the human body? So as you can see here, as the fetus later develop further, what we can see is that because the ectoderm will fold, ectoderm will have a faster proliferations of the cell, so it will fold because it's longer, so it's gonna fold itself. While the mesoderm will follow, so this is the heart, and this is the blood vessels, and you have the stalk that connects with the placenta, and then you have the endoderm here, which will form the pharynx, larynx, GITs, and also the bladder. So what happened is that we have this yolk sac here, but since the growth of the yolk of the endoderm is slower as compared to the ectoderm, so we need the vitelline and also the yolk sac that which pinched off from the embryo. But we still need this space because we need a space for the rotations of the GITs of the colon and also the small bowel. So we need this, and later on it's gonna pinched off. But if it's persists, then it's become this is the vitelline. So it will become the macrodaverticulum. This is a question that previously asked. So the macrodaverticulum is 2-2-2 principle. So 2% of population had this, 2 feet from the ileocecal junctions, and another 2. I can't remember another 2. So basically this is what happened.